are going to look at the structural features of amino acids. And amino acids are the building blocks of proteins. Proteins are vitally important in terms of the infrastructure of a cell or of your body. They also are the functional parts of enzymes, and enzymes are the little machines that allow us to metabolize our food, to build new strands of DNA, and to do a host of other things in the cell. In Classroom, you will find a copy of the guided notes, and if you could print those out and then fill the guided notes as we move through the presentation, that would be ideal. You can take a picture of your guided notes and then just send them to me through email or through Classroom. If you can't print your guided notes, I will make some copies available at a future date. This presentation is about amino acids and proteins, and amino acids are the building blocks of most of the parts in the cells of our body and our organs. There are 20 alpha amino acids, which are the building blocks of the proteins in living cells, and 8 of those amino acids are considered essential amino acids. So the cells of our body cannot synthesize those amino acids. Therefore, they have to be provided in our diet. So our bodies are comprised of cells, and cells are made up, at least in part, of proteins and amino acids. The proteins that are constructed in cells are assembled in a protein factory within the cell called the endoplasmic reticulum. The endoplasmic reticulum is just like a large workbench, and upon the surface there are ribosomes and other components that are needed to build proteins. So muscle, hair, and fingernails are all examples of parts of our body that are almost completely all protein. So the essential amino acids and other amino acids can be supplied in our diet. Fish, meat, and dairy products are high in proteins. Our immune system depends upon proteins to construct parts like antibodies, and the antibodies are the part of our immune system that attach to invading viruses and bacteria and mark them for destruction by white blood cells. Sometimes the antibodies themselves can destroy an invading pathogen. Viruses are dependent upon proteins to infect cells and to replicate themselves. The COVID-19 virus involves the use of several different very important proteins for it to complete its infection cycle. So this is a diagram of the COVID-19 virus. There are spike glycoproteins along the surface of that virus that enable the virus to attach itself to the membrane of the host organism. That starts the infection cycle. There are hemagglutinin esterase spikes also along the surface. Those really are enzymes that have several different functions in the infection cycle. They do help the virus to attach to the membrane of the host cell. They also make the membrane permeable so that the RNA molecules can be inserted into the cytoplasm of the host. Once the new viral particles have been assembled by the host cell, these spikes help the new virus particles break free from the cell membrane, go on to infect other cells. There are membrane glycoproteins that interfere with antibodies that the host organism might produce to attempt to attack the virus. The nucleocapsid proteins surround and protect the virus RNA and are essential to duplicating the RNA within the host cell. Again, proteins are vital to construction of our organs, our red blood cells, our immune system, and the parts of the cell that keep us alive. Some facts about the proteins in your body. Each cell in your body contains 9,000 different proteins. The human body contains over 100,000 different proteins. That includes the enzymes that are needed to repair and copy DNA. So at this point, go ahead and stop the video. See if you can summarize to yourself some of the important things about amino acids and proteins that we've talked about. So there are some basic features that are common to all amino acids. All amino acids contain a carbonyl. You might remember from organic chemistry that a carbonyl is a functional group consisting of a carbon atom double bonded to an oxygen atom. And in these models, red represents oxygen, gray represents carbon atoms, white represents hydrogen atoms, and then blue represents nitrogen atoms. So all amino acids also have an acid group, and that acid group is called a carboxyl. Again, a carboxyl is an OH group attached to a carbonyl. So this entire structure then would be considered a carboxyl. Amino acids all have an amine group, and the amine group consists of a nitrogen atom bonded to hydrogen atoms or carbon atoms. These are called alpha amino acids because they have an alpha carbon. The alpha carbon is that point of attachment for all of the other functional groups in the amino acid. So notice in this case that the amine group and the carboxyl group are both attached to the alpha carbon. Then the feature that makes all amino acids unique, or makes them different from each other, is the R group. And R groups can be a lot of different things. They can be additional amine groups, additional carboxyl groups, or other organic structures. So some examples of amino acids. The very simplest amino acid is glycine, and in the glycine molecule, the R group is simply a hydrogen atom. The amino acid proline consists of an R group of carbon atoms that then fold back around and attach to the amine group of the amino acid. Phenylalanine contains a benzene ring, and oftentimes there is an OH group, hydroxyl, attached in the para position on the benzene ring. Cysteine is an amino acid that contains sulfur atoms. Those sulfur atoms can cross-link with other cysteine amino acids in the polypeptide chain, forming what are called disulfide bonds. If you have curly hair, you have a lot of disulfide bonds in the keratin proteins of your hair. Valine is an amino acid with an isopropyl group attached to the R position. Amino acids are also optically active, just like sugars. So there is an L-alanine and a D-alanine. Those molecules are mirror image isomers. So at this point, pause the video again, see if you can summarize to yourself the important structural features of amino acids. So proteins then are long chains of amino acids. You might be wondering how a protein forms from amino acids. Well, two amino acids link together to form what is called a peptide bond. Those linkages occur because of attraction that takes place between positive and negative charges. The amine group of one amino acid has an unshared pair of electrons, and the carbonyl carbon of a second amino acid is positively charged. That happens because the oxygen atoms take the electrons more than their fair share of the time. So then attraction between positive and negative charges, or a nucleophile and an electrophile, cause these two amino acids to link. The carbonyl of one amino acid will always link to the amine group of a second amino acid. Water is split out and a peptide bond forms. So proteins are long chains of amino acids with varying side chains, or R groups, that give the protein unique properties. And interactions between side groups then give proteins specific three-dimensional shapes. So there are levels of structure to proteins, and each level is very important to the behavior or the function of that protein within a cell. So the first level of structure is called primary structure, and it's determined simply by the order or identity of the amino acids in the polypeptide. Secondary structure can result in the formation of two different possibilities. There is the helix, which is coiled or spiral in shape. Then there is what we call the strand or the pleated sheet, which is a wavy or folded pattern that takes place within the structure of the protein. Tertiary structure gives the protein its three-dimensional shape, and tertiary structure is determined primarily as the R groups on amino acids interact with each other. Last of all, there is the quaternary structure. And quaternary structure occurs when several different proteins come together and form a functional group. So again, pause the video and summarize for yourself the different levels of structure found in proteins and what effect those structures have on the protein.